So yes, unlike Vegas, what happens in the liver doesn't always stay in the liver. We got the nitrogen to the liver. Let's get it out now. Okay, and so that's where the next one will come into play. Okay, part 2. So this should look familiar. It's what we've done, what we've already talked about. And so once again, make sure you understand it. Just to do a quick review, if you're going to move nitrogens around in the same cell, so intracellularly, you just want to move a nitrogen from one place to another place, what amino acid do we typically use? Does the cell typically use? Glutamate. If you're moving it within the blood, you're going to be using either glutamine or alanine, depending upon the tissue of origin. Okay, skeletal muscles, typically, especially if they're in use, will use alanine. Everything else, including skeletal muscles, not used, but you know, if they're in relatively resting mode, can utilize glutamine or will utilize glutamine. Okay, and so ultimately, once it comes in here, we have this glutamate intermediate, you know, from the liver itself that's going through with the aminotransferases that we've seen, and then we're going to have to try to get rid of its nitrogen as well. Okay, and so one way is that, well, we'll talk about, there's glutaminase that can also remove the nitrogen. Okay. Let's see where we're going now. So, since I just talked about the uricotelic and ureatelic and all that kind of stuff, then we're not going to show that same slide again. Okay. So, for ureatelic organisms, we use the urea cycle to ultimately make urea. There are 5 steps. It starts in the liver and mitochondria, but then 3 of the steps take place in the cytosol. So that's the reason why I say it's more like gluconeogenesis in that sense, with respect to Partknowization. Okay. We've actually seen a little bit of it before. This is what it looks like overall. So that's why I kind of like this in the sense that we can see where parts of it are in the cytosol. There's the hepatocytes, mitochondria that does a portion of it, and then it sticks it back out to the cytosol in order to ultimately get the urea, which the urea then can be released. Okay. So what we're going to be doing next, and you guys don't have this slide yet, is I would expect you to understand already, based off of today's lecture, as well as the stuff that we've done in the past, where the enzymes and the precursors came from. And you should start to recognize what some of these precursors are. Rather than get starting on it, you know, we have like 5 minutes, I think that's a good time to just end this. It's alanine, it's coming from skeletal muscles. On the other side, what is this over here? That's a nitrogen that's kind of hidden by that. This is a nitrogen, but it's glutamine, and it's coming from intercellular. Yeah, it can literally come from any of the tissues. It's just that all non-skeletal muscle tissues have to use glutamine. And so, yeah. Okay. And this, let me just point it out. This right here, what enzyme is this showing right here? The amino acid becoming this, and we're using alpha-huglutarate. So what is this enzyme? It's just a different way of showing it. It is. It's an aminotransferase. Okay, it's just another way to show it. And so, for example, here, this amino acid is what's going with this alpha-keto acid. It's just a generic alpha-keto acid. So alpha-ketoglutarate is becoming, what's this right here? Glutamate. Whereas if you're having alanine, then the alpha-keto acid is specifically pyruvate. They're just trying to show it as a generic overall. But that, yes, that is glutamate, and it is made from an aminotransferase. Okay. Let me see. Okay, so unfortunately this is all one slide. So what enzyme transforms glutamine and breaks it up into glutamate and ammonium? What enzyme is that that's over here? So it's glutamine, it's one enzyme, and they're showing you
there are two products. There's ammonium or ammonia, and there's glutamate. So how do you get glutamine from glutamate?
Glutamine synthetase.
Pardon?
Glutamine synthetase.
Okay.
Backwards.
Glutamate.
Right? If we're going backwards, yes. But this one's glutaminase. Okay, whenever we're doing specifically the breakdown, okay, within this liver hepatocyte mitochondria. Okay. So here we're breaking it down to make glutamate and to free up the ammonium. And that way, if we had shown it going backwards at the expense of ATP, then it's going to be synthetase.
Is it the same enzyme when it goes, like, or, you know what I mean? Because it's pretty much the same.
I don't know. I don't think so. I don't think so because, well, sometimes I always say absolutely no because then we have that weird, FBPase1, FBPase2, PFK2, where it's really the same, but it's the different sides. And so I don't think glutaminase and glutamate synthetase is like that. I think that these are two separate ones because this does not require ATP, whereas to go backwards it is. It's oxidative deamination. All right, so we have glutamate, we have ammonium. Now we've got, let's see, I'm gonna fix this one of these days to where I don't do more than one box at one time. Now we have to figure out what, so that's one way to get glutamate and ammonium. The ammonium from the glutamine, we'll just go ahead and intron in, okay? But we still, at some point in time, we want to get rid of this glutamate. And so to get rid of the glutamate, we can also get rid of its nitrogen. So then we have to start to think about what we can do there. So what enzyme is each of these boxes? And it doesn't matter to me where we start with because they're on the same slide. So the way to read this is glutamate. If it loses its ammonium, it's going to be alpha-ketoglutarate. Or if glutamate goes with this enzyme right here, where it hooks up between aspartate and oxaloacetate, it's also, we have alpha-ketoglutarate, okay? So that's why we have two possibilities here. It doesn't matter to me which one that you want to pick. You want to pick this first one, or do you want to pick the second one?
The first one.
Okay, the first one, what enzyme is that first one? What enzyme can take glutamate and remove its ammonium without hooking up with this or something akin to this? Remember, we cannot use an aminotransferase, so otherwise you're just going to be making another amino acid over and over and over and over again. It's what? It's the dehydrogenase. And which one specifically is it? Glutamate dehydrogenase. They just didn't show in the NADH. Which is a shame. If they had done that, it'd be really easy. You'd say, oh, look, it's glutamate dehydrogenase. But yes, that first one is glutamate dehydrogenase. That's how glutamate gets rid of its nitrogen ultimately in order for it to enter in the urea cycle. Okay, on the other side, and there's a reason why they do this, is because aspartate plays a role in the urea cycle. So this is another way to get aspartate. So how do we get aspartate from oxaloacetate at this expense of a glutamate as well? What amino acid? That doesn't make sense. What enzyme is that? Is what? It's aminotransferase. Now this one we can say what specific aminotransferase it is going to be. It's aspartate aminotransferase, okay? So in order for us to utilize all of this urea cycle, it takes more than one glutamate. Okay. So we can use one glutamate in order to make this aspartate. Or you can get aspartate from someplace else. We can use this other glutamate, this other one. We can use another glutamate in order to get its nitrogen in order to go into the urea cycle. Just like we can use this glutamine to get the glutamate and the nitrogen. So that's why you can have multiple sources, potential sources of glutamate. They're just trying to show you all the possibilities here. Okay. This is where you know how we had a step 0 for the citric acid cycle? This is step 0, if you want to think of it that way, for the urea cycle. Because it's not technically a part of the urea cycle yet. We can't take, for us mammals, we cannot take ammonia, or ammonium in this instance here, and just stick it directly into the citric acid cycle. Just like we couldn't take pyruvate and just stick it directly in citric acid I think I said it wrong. Okay, we can't take ammonia and stick it directly into the urea cycle, just like we can't take pyruvate and stick it directly into the CAC cycle. We had to transform it first. All right. This enzyme is what's going to do that. It takes ammonium and it makes carbamoyl phosphate, okay, which the carbamoyl group is probably not one that you learned in organic chemistry. I used to not teach it in organic chemistry. That's like a, you know how the acyls is a carbonyl group. The carbamoyl group is this. It's an amide that doesn't have an R group hanging off of it. Okay, so it's not used as that often. Probably in organic chemistry too. Okay, but that's how it's the thing. That's carbamoyl phosphate. Right. So what type of enzyme is that? Look at everything involved. We have ammonium. We have bicarbonate. This is the way it's, we have ATP. We get back two ADPs and we only get back one inorganic phosphate because the second one is actually stuck to make the carbamoyl phosphate. That's why they've got this color-coded. We have the nitrogen that came from the reason why, the whole purpose is to get rid of the nitrogen. We've got one of the phosphates that came from the ATP. And then we've got the carbon that came from the bicarbonate, which we have plenty of that around. You know, that's, if you remember way back, we even talked about it last semester, carbonate, the bicarbonate is actually made by carbonic anhydrase from carbon dioxide. So it's part of the respiration pathway. So what class of enzymes can that be? It is a synthetase. Okay, so if you're going to name it, oh, and well, first of all, why do we know it's a synthetase? It uses ATP. So if you're going to name it, what would you name this one? Right, in particular, it's carbamoyl phosphate synthetase, and it's isoform 1. But if you just said carbamoyl phosphate synthetase, I'd give it to you. Because offhand, I don't even know about carbamoyl synthetase. Carbamoyl phosphate synthetase, isoform 2 does, off the top of my head. Okay. Any questions there? Make it smaller, where we can see it all. Okay. All right. So now look, we're still in the mitochondrial matrix. We're getting close to leaving though. The official first step of the urea cycle is this one. It takes ornithine, and we have not seen ornithine before. First of all, does anyone know where does the name ornithine come from? What is its root? What is ornithology? Quentin? I figured if anyone knows, it'd probably be you. What is an ornithologist? What does he or she study? Birds. Okay, I don't know historically. It's just one of those facts that it's stuck. But it comes from bird. I mean, it's named after birds, but that's an actual alpha amino acid. Remember how I told you that I made you memorize the 20 naturally occurring amino acids, and then later on I kind of ventured to say that are used to make proteins. Those are the ones you had to memorize. This is another one. This is an alpha amino acid. Because look, we have a carboxylic acid, there's a C alpha, and there's an amino group. It's just, this one is not used to make proteins. It's used in the urea cycle. Okay, but otherwise, it's an alpha amino acid. All right, so it takes ornithine, it takes a carbamoyl phosphate, and it smooshes them together. You can't see it, it's inorganic phosphate that's leaving, to make citrulline, which is also another, and it's naturally occurring alpha amino acid. It's just, once again, not used in production of making proteins. Okay. And I think this is the first time that we've seen that. We'll see other examples later on.
What else can citrulline be used for?
What else can it be used for?
Yeah, I just see it in ingredients sometimes.
In humans, I don't. Well, I mean, well, I don't want to give it away. I guess ultimately you can use it to make arginine if you really wanted to. It's probably not the most. We're gonna see. Arginine plays a role in a moment, but in humans, I can't think of anything in particular that we would use it for. It's not to say that it's named after citrus fruits. It is naturally occurring, and so it's probably other species and organisms use it for various things that I know of. Except for this way. I guess if you use it, it can help you get rid of nitrogen as well. since it's bringing more nitrogens in. So I don't know. Maybe it'll help jumpstart the nitrogen process. But yeah, so it probably wouldn't hurt you in that sense because it's just nitrogen. Okay. All right. So this enzyme has got a horrible. Well, for me, it has a horrible name for me to say. I have problems saying this name from all my years of speech therapy. You probably won't have any problems with it. But and that's why I say it has a difficult name. Whenever you see it, it makes sense because of what it's doing, but I have problems saying it. So what is this doing? Like if I was naming it, I know I would name it, but they didn't ask me. So it's not a synthase. Like that's what I would name it. I'd be like, oh, this is synthase, right? It's not. What else is happening here? We are adding, but it's not a carboxyl group. What are we adding? What are we transferring? It is an amide, and that amide, just like we have acetyl means it's only got two, this is a carbamoyl group, which is hard for me to say. So we are transferring a carbamoyl group. And we are transferring it to ornithine. And so its name, oh whoops, got cut off. Its name is ornithine transcarbamoylase, which I feel like I'm going to swallow my tongue every time I say it. It's hard. It's that. I have probably seen carbamoyl to begin with. I don't know why. But yes, it's that. Which makes sense now, because it is transferring that, which is a carbamoyl group, to ornithine. All right. Now we can leave the mitochondria. Are we doing on time? Good. So one thing I want to point out, there's this aspartate. Aspartate can go back and forth. We've seen that before. It can be shuttled back and forth. I didn't see it before. With respect to making oxaloacetate and things like that. And so it can just leave. And it plays a role later on in a different step. Citrulline can leave. Ornithine can come back and forth as well. All right, so now this is the second part of the urea cycle, which happens in the cytosol. This very first, the first enzyme was ornithine transcarbamoylase. This next enzyme is a two-step process, which makes it intermediate. That's why they're showing it that way. So we take the citrulline, we utilize an ATP. It makes this citrulyl AMP intermediate, which is this really good leaving group, in order to ultimately, and it hooks up with aspartate. See, the aspartate that was from before comes back down, knocks off the AMP in order to make argininosuccinate. All right, so that they've got it labeled as 2A and 2B. But we do utilize an ATP. It utilizes an aspartate from previously. And they've got it color-coded, so that way we can see this is the aspartate. This is all the, from the citrulline originally. Okay, and this is the nitrogen that we've been trying to get rid of all along. Well, actually, we're going to be getting rid of both nitrogens, but this is the original nitrogen that we had from before. So what class or classes of enzymes can do this? Pardon? Okay, would it be a synthase? What do you think? I thought I heard someone say something under the breath, and hopefully it was something positive. I don't know, all the soggy recycle always makes me want to go pee. But I mean, it's going to water, running water. It's a synthetase, and we know it's a synthetase because ATP. All right, and what are we synthesizing? What are we making? Pardon? Argininosuccinate. Yes, if you can get that to fit to like a song, that'd be awesome. Argininosuccinate. So this really is argininosuccinate synthetase. So its name makes perfectly logical sense. So then the third step, I'll give you a moment to write that aloud because, listen, for those of you who are writing, argininosuccinate. synthetase, which we've seen succinate and its derivatives where? What other metabolic citric acid cycle? That's the reason why, hopefully we'll see it makes it a little bit more obvious that the citric acid cycle, the urea cycle really are linked together, and they used to call it the bicycle. Why isn't it a bicycle? You don't say, I'm going to go ride my bicycle. No, bicycle. I'm going to stay here and call it bicycle. Okay. Okay. So now the third step is as follows. Oh, this drives me nuts. So this third step is we take this argininosuccinate, and what it's going to do is it's going to cleave it or cut it to where those that is that pink comes off, and that's fumarate. Right? And so before we go any further, where can fumarate go? Citric acid cycle, all right? And then this is just arginine, all right? That's why when you asked about citrulline, I'm like, I guess you can use citrulline to make arginine. Typically, that's probably not going to be a major.
If it's for, you can make nitric oxide with it.
Mm. Well, yeah, because an arginine is used for making nitric oxide as well. It's
Makes your blood pump.
Mm-hmm. But yeah, it's just not a. Yeah, it's not something that we. It's something that we can make. It's not something you have to take externally from your diet. You don't have to worry about, I can make my citrulline come, quota. All right. So what class of enzymes can this be? Oh, can do this. This one actually does have a unique name. We've seen some of those unique names before. So if you're going to give it a unique name, but we have a fumarate already. That's true. But you're on the right path. Well, what? Well, fumarase was with fumarate. So just think of one of those analogies, if this was like the SAT. Fumarase is the fumarate as blank as to argininosuccinate. Thank you, it's argininosuccinase. Yeah. That's a great, GRE kind of thing, like this blank is. All right. And then, so I didn't hit the next enter. Then the last step also has a very special name as well. But it's one where the arginine is hydrolyzed by water, and it kicks off the urea, and then we're left with ornithine, which then go back on it there for ornithine transcarbamylase to repeat the urea cycle. And so knowing how that they have these special names, what would you call it? What?
It's not arginase, is it?
You're very, very, very close. If you had written that out, you would see it. Because, you think, arginase, but they make it a little bit easier. It's arginase. But, yep. So are there any questions on the urea cycle? Once again, it's bicompartmentalized. It's part of the bicycle, which that's what the next slide's about. All right, so the urea cycle and the citric acid cycle are linked. It's called the aspartate argininosuccinate shunt. Okay, and so make sure you just understand this. It looks really complicated, but it makes sense. We've actually seen all this kind of stuff before. This is what we've just been talking about, the urea cycle, which we get this citrulline argininosuccinate. Argininosuccinate makes fumarate, which fumarate then can enter into the citric acid cycle. As a part of the citric acid cycle, it can form malate, right? And because of that, malate can go in and out as well. And so malate can be part of the citric acid cycle, or malate can also help feed into this if need be, which it doesn't here. But Likewise, we can make aspartate from what's missing there. If you were going to write this in, what did the aspartate come from? It's oxaloacetate. Okay, and so the oxaloacetate can make it. And that's why it's called the aspartate argininosuccinate shunt, because it's another way to siphon off from the citric acid cycle. You know, when you don't need the nitrogens anymore and you don't need the energy, then you're going to want to get rid of it. Okay.
Are there equal concentrations of like fumarate, malate, in like the cytosol or in the mitochondrial matrix, or is it based upon what your cell needs? And so what are those cases?
I don't know for certain. I would say it's based off of your cellular needs, and it's probably also going to be based off of what tissue we're talking about, right? Because this is occurring in the liver, whereas there may be completely different concentrations in skeletal muscles, and it's very different energetic needs in skeletal muscles. No one's saying that would be a hypothesis. I don't know the actual concentrations. Any other questions? Urea cycle is not too difficult. Just, remember that it's got two special names, and it's got that really hard one to say. Ornithine is not hard, but trans-carbamoylase, which is transferring carbamoyl to ornithine. Okay. Any questions? Awesome.